Hello everyone, a very good morning. Today let's discuss on our next basic science topic, that is stool examination of parasites. So in stool examination of parasites, I've taken this topic from our Panikka's parasitology textbook. Okay, here you have a microscopic examination, next will be your concentration method, and third will be your egg counting methods. I've made this topic as small as possible. Okay, if you remember these points, it is enough for your exams, right? Coming to the microscopic examination, we have three methods here. So there are three methods in your microscopic examination. First one is your wet mount, second one is your thick smear, and third one is your permanent stained smear. So one is your wet smear or wet mount, next one will be your thick smear, third one will be your permanent stained smear. Yes, coming to the wet mount first. In wet mount we have four types. So four types of wet mount can be done. First one is your unstained wet mount. So in an unstained wet mount or wet film, here we use a drop of the stool, a small quantity of stool with a drop of a saline is being used in the slide. Then after placing that to, you are placing a cover slip, applying a cover slip. Then it is examined under your low power microscope, that is called as your unstained wet film. That is small quantity of stool in a drop of saline. That saline is your 0.9% saline. Okay, that is placed on the slide, and you apply a cover slip. Then after this, then you will examine this under low power microscope. Low power is your 10x. Okay, yes. Next, second one is called as wet saline mount. Second one is your, this is first one, second one is your wet saline mount. In wet saline mount, it is used mainly for detecting the live motile trophozoites. So it is mainly used for detecting live motile trophozoites, and eggs of the helminths are seen in this. Eggs of helminths are seen easily from wet saline mount. Third one is your eosin stain. This all comes under your wet mount only. Okay, third one is your eosin staining. So in eosin staining, you use 1% of eosin aqueous solution. So you use eosin 1% aqueous solution. So this indicates the viability of the cyst. It is mainly for viability of cyst. How do they identify the viability of the cyst in this is, live cysts are unstained while dead ones will be stained with your eosin. So live cysts are unstained while your dead one will be stained with a eosin, or stained with your eosin pink. Okay, so this is eosin cyst, mainly for viability of the cyst. The fourth one will be your iodine staining. So in iodine staining, it helps to confirm the identity of the cyst. It is whether it belongs to which group of organism, whether it is helminths or protozoal organism. Okay, so it helps to confirm the identity of your cyst, right? Yes. Coming to the next one. Next method is your thick smear method. This is not useful for your routine examination, but it is valuable in survey for the intestinal helminthal X. It is not useful in routine examination. That one you have to mention. So not useful in routine examination, right? Next, it is mainly for the survey of intestinal helminthal X. Okay, next, this is also not useful for identifying larva. Protozoal or helminthic larva is not useful for diagnosis of larva. Okay, next, permanent stained smear. It is mainly used for confirmation of intestinal protozoa. Both your tropozoites and cyst can be identified by your permanent stained smear. So in permanent stained smear, it is mostly used for confirmation of your intestinal protozoa, both your tropozoites and cyst. Okay, yes, here you have two methods. Main two method of your permanent stain are, one is your iron hematoxylin stain, second one is your Wetleys stain, Wetleys trichrome stain. Okay, so one is iron hematoxylin stain, second one is your Wetleys trichrome stain. These are the two methods that is used in your permanent stain smear. So coming to first one, that is your microscopic examination. You have wet mount, thick smear and permanent stain smear. In wet mount you have 4. That is your unstained wet film, wet saline mount. Next will be your eosin, and fourth one will be your iodine staining. Well, coming to the thick smear, it is not done routinely for your examination. While permanent stained smear, it is mainly used for confirmation. These are the important key points you have to note down for this question, right? Yes. Coming to the second method, that is your concentration method. This concentration method, for what we use concentration method? Only when the parasites are very scanty in the stool or for routine examination fail to detect. So when the parasites are very less in number in the stool, then you go to, then we'll use a concentration method to detect them, because while doing a routine examination you can't find the parasites there. So only when parasites are scanty in stool, we use concentration method. So it is necessary to concentrate protozoan cyst and helminthal eggs and larva for identifying the species. So the techniques that is being used in your concentration method are, one is your flotation method, and second one is your sedimentation method. First one, in your flotation method, here your fecal sample, it's just your stool sample, is suspended in a solution with a high specific gravity, so that your parasite eggs and your cyst will float in the top. Okay, so in a flotation method, the fecal samples suspended in solution with high specific gravity, so your cyst and the eggs, this will float at the top, so that they can separate the cyst and eggs and do a normal routine examination. Okay, this is flotation method. While coming to a sedimentation method, it is just opposite of that. That is, your fecus is suspended, that is your stool sample is being suspended in a solution with low specific gravity, so that your cyst and eggs are sedimented at the bottom, either by spontaneously or by using a centrifugation. Your eggs and cyst will sediment at the bottom. Then after that you will do a this thing what is called routine examination in the slide to look for the eggs and cyst. So here ecus is suspended in solution with low specific gravity. Okay, so that your cyst and egg will sediment at the bottom. These are the two techniques that is being used in your concentration method. One is your flotation, another one is your sedimentation method. Right? Next is your egg counting methods. I have not gone into a detail of your egg counting methods. I have just written the techniques that's being used for your egg counting methods. Techniques that is being used for your egg counting methods are: first one is standard wet mount. Second one is your thick smear technique. And third one is your Stool's dilution technique. Okay, so first one is your standard wet mount, second one is your thick smear technique, and third one is your Stool's dilution technique. These are the techniques that is being used for the counting eggs that is being present in the stool sample. Okay, so these are the main points that you have to write for the question if they asked stool examination of parasites. Thank you for subscribing our YouTube channel. Let me come up with another video of basic science tomorrow. Thank you.